This video is going to be about astaxanthin. It can be found in salmon. So basically, it's a reactive oxygen species suppressor, and it's in the mitochondria and localized carotenoid antioxidant that quenches lipid peroxyl radicals, especially in membranes. It also protects the cardiolipin and mitochondrial ETC. Now the cardiolipin is inside the mitochondria, and it has to do with the cytosol. So basically, if the cytosol escapes, or I mean, if so basically it's supposed to prevent unnecessary apoptosis. So if astaxanthin protects the cardiolipin, that can actually support the prevention of unnecessary apoptosis, which wouldn't be good, because then that would actually be damaging to tissues, or even autoimmune related. So astaxanthin from salmon could actually prevent autoimmune disease. So the key results would be reduced lipid peroxidation and less oxidative protein misfolding. Astaxanthin also protects immune cells in the tumor microenvironment, because when the extracellular matrix opens, the immune cells have to go in to attack the cancer cells, but they need a shield of protection, and astaxanthin can provide that shield. Like macrophages, NK cells, CD8 T cells are exposed to high reactive oxygen species, oxidized lipids, also hypoxia, which actually leads to mitochondrial failure. If the mitochondria is reduced, then that means less ATP, less energy for immune cells. So astaxanthin protects from hypoxia as well, because cancer cells thrive on low oxygen environments. Astaxanthin preserves the mitochondrial membrane potential, maintains ATP, prevents T cell exhaustion, because astaxanthin acts as a shield for immune cells. Also, astaxanthin also works against CD47 SERPA signaling. So basically, that means excessive reactive oxygen species reduces the calreticulin exposure on the cancer cells. So basically, if the CRT cannot expose on the cancer cell, then the macrophage can't do the phagocytosis or the engulfing. So astaxanthin reduces the reactive oxygen species that would otherwise lead to the oxidation of the CRT. Because if the CRT is oxidized, then the immune cell can't do the phagocytosis. So with astaxanthin, the lipid oxidation is reduced, which means that the CRT, or that's short for calreticulin, can be preserved on tumor cells, so that the CD91 on the macrophage can bind to the CRT on the tumor cell, creating an engulfment or a phagocytosis. Reactive oxygen species reduction weakens the CD47 dominance on the macrophage, because reactive oxygen species causes oxidized CRT on the cancer cell, which means that the CRT cannot bind on the CD91 on the macrophage. Also, with reactive oxygen species reduced, that means that the CD47 on the macrophage is weakened, which means that the cancer cell cannot bind onto the SERP alpha on the macrophage. Or basically, the CD47 on the cancer cell is reduced by weakening reactive oxygen species, because the CD47 on the cancer cell cannot bind onto the SERP alpha on the macrophage. So basically, reducing reactive oxygen species with astaxanthin can stop the cancer cell from binding to the SERPA. So basically, tumors try to exploit this pathway called the HSP70 GP96 bind tumor peptides. The calreticulin is exposed on the tumor cell, and then the CRT binds to the CD91 on macrophages, and then that leads to phagocytosis. But the tumors will try to escape that by reprogramming lipid metabolism, and also through oxidized phospholipids, or OXPLS for short, that accumulate in the membrane. And as a result, the HSP70 and GP96 misfold or lose peptide loading efficiency. What that does is that it impairs the CRT translocation to the plasma membrane. So what that means is that the cancer tumor cell actually prevents the CRT from showing up on the outside of the membrane where it's supposed to be. Instead, it's on the inside. And if it's on the inside, that means that the macrophage cannot do phagocytosis if the CRT is on the inside. So basically, tumors know how to escape that by intentionally bypassing the HSP70 and GP96 pathway, which pretty much means that antigen presentation collapses. That means that the macrophage cannot see the cancer cell to attack it. So basically, the CRT is down-regulated, and oxidized CRT has reduced CD91 affinity, and surface CRT density drops. Basically, that the CRT on the cancer cell is not working. So tumors also upregulate CD47, which is an oxidized stressor that stabilizes CD47 on the membrane. And also, tumors also engage with the SERP alpha on the macrophage with the CD47. What that does is that phagocytosis, the engulfing, is actually reduced or inhibited because the tumor did that with the CD47. But basically, salmon with its astaxanthin can actually prevent or at least try to stop the CD47 on the cancer cell, because astaxanthin quenches the oxygen and lipid radicals and also prevents OXPL accumulation in the ER membranes. Cancer tumors will try to upregulate OXPL oxidation. That's the thing that actually that pulls the CRT into the cancer cell, so that the CRT is not on the outside. So that means that the macrophage cannot do phagocytosis. But basically, salmon with this astaxanthin stops that from happening by reducing the oxidized PL that the cancer cell uses to hide the CRT so that the macrophage can't see it. So basically, that means that astaxanthin actually strengthens the eat me signal that the cancer tumor is trying to hide from the macrophage. So without astaxanthin, lipid oxidation leads to the HSP70 GP96 failure, which actually favors the tumor, which leads to the CRT going down, and then the CD47 on the cancer cell thrives, which is what the cancer cell needs in the absence of astaxanthin, which leads to immune escape. But with astaxanthin, all those things are disabled. Please subscribe, like. I've got more videos coming up, and I'll see you in the next one.